Okay, good afternoon, students. Now let us continue with the chemistry of heme. So now I am going to explain. We are going to discuss about hemoglobinopathies. Hemoglobinopathies are a group of disorders which are due to mutations in the genes which are responsible for synthesis of alpha and beta chains. Hemoglobinopathies are a group of disorders characterized by or due to mutations in the genes which are responsible for synthesis of globin chains of hemoglobin, that is alpha and beta. Because of this mutations of the genes, what happened? There is change in the amino acid sequence. So change in the amino acid sequence that would result in the synthesis of abnormal hemoglobins. So normally if you remember the structure of hemoglobin, heme plus globin to form hemoglobin. There are four types of globin chains are present, 2 alpha, 2 beta. Now here, these hemoglobinopathies are a group of disorders due to change in amino acid sequence of globin chains. Why amino acid sequence has been changed? It means because of mutations of the genes which are responsible for synthesis of globin chains. So because of mutations of globin chains, there is amino acid sequence change that would result in the synthesis of abnormal hemoglobins. Now here, these abnormal hemoglobins are the resultant of mutations already we have discussed. These abnormal hemoglobins are the resultant of mutations in genes that code for alpha and beta chains of globin. Okay, because of mutations of alpha and beta globin chains, there is abnormal formation of hemoglobins have been identified. 400 mutant hemoglobins are known till now, but 95% of the abnormal hemoglobins are because of single amino acid alteration in the single amino acid of globin. Okay. Nearly 400 different types of mutant hemoglobins have been identified, but 95% of all hemoglobin variants are due to change in single amino acid sequence of globin chain. Now, to understand these hemoglobinopathies, you should have knowledge about globin synthesis. Okay, you should know about basics of globin synthesis. So, globin chains are organized into two gene families: alpha gene family, beta gene family. Alpha gene family means here two genes are there on chromosome number 16. Okay, see here, now we are discussing about globin synthesis to understand hemoglobinopathies. So globin genes are categorized. Globin chains are organized into two gene families like alpha, beta. Alpha genes are located on chromosome number 16. Alpha genes are located on chromosome number 16. So there are two genes are there on chromosome number 16 which codes for alpha globin. And along with that, one zeta gene is also located at chromosome 16 that is active during embryonic development. Okay, so on chromosome number 16, two genes are there. Okay, two genes are there which encodes for the synthesis of alpha globins, and one zeta gene is there that is active during embryonic life, that is also located on chromosome number 16. Next one, beta gene family. So here, beta gene family, a synthesis of beta chain occurs. This beta gene families are located on chromosome number 11. So there is one gene is there on chromosome number 11 responsible for synthesis of two beta chains. What about alpha chains? But for the synthesis of two alpha chains, two genes are located on chromosome number 11. But for the formation of two beta chains of hemoglobin, only one gene is responsible, that is located on chromosome number 11. And even chromosome number 11 also contains, along with the beta gene, it also contains one epsilon gene, two gamma genes, and one delta gene. One epsilon gene that is expressed active in early stages of embryonic development, and gamma genes which could able to synthesize gamma chains. Gamma chains are present in structure of fetal hemoglobin. As you know, fetal hemoglobin is composed of 2 alpha and 2 gamma. and delta gene, and this delta gene is also producing delta globin chain found in adults. Okay, adult hemoglobin A1, if you know the compositions, hemoglobin A2 is composed of 2 alpha and 2 delta. So that delta gene is located on chromosome number 11. Fetal hemoglobin composed of 2 alpha and 2 gamma. That gamma genes are located, the genes which help in synthesis of, which can synthesize gamma globin genes located on chromosome number 11. And even epsilon gene that is there that is seen in embryonic development is also located on beta gene family. Now here, this is the organization of the globin genes, and this is alpha and beta. Okay, this is chromosome number 11. So this is chromosome number 11 and 16. So genes responsible for synthesis of 2 alpha globin chains is located on chromosome number 16. Okay, 2 alpha 1 and alpha 2 genes are responsible for synthesis of 2 globin chains, 2 alpha globin chains, which are located on chromosome number 16. Along with that, there is zeta. Okay, there is zeta globin gene is also present on chromosome 16, which is active during embryonic life. Now here, this is chromosome number 11. So on chromosome number 11, single gene which is located in chromosome number 11, beta gene, that is responsible for the formation of 2 beta chains, and along with that it also contains one one gamma, delta, and one delta gene is there that is responsible for the formation of hemoglobin A2, since it is composed of 2 alpha and 2 delta, and there are 2 gamma genes are there which are responsible for synthesis of fetal hemoglobin. And even along with this, it also contains epsilon gene. So this gene active during embryonic life. If you remember the synthesis of the fetal hemoglobin, during first 6 months of gestation, first hemoglobin is composed of 2 zeta and 2 epsilon. And within first 6 months of pregnancy, this epsilon and zeta has been replaced by 2 alpha and 2 gamma. So at the time of the birth, 80% are of hemoglobin F type, and within first 6 months, see these globin alpha 2 remains like that, gamma 2 has been replaced by beta. So that will convert HbF to HbA. Okay, fetal hemoglobin has been converted to adult hemoglobin. So anyway, so the genes responsible for the formation of, for the synthesis of alpha chains are located on chromosome number 16. The genes which are responsible for the formation of beta chains, delta chains, gamma chains are located on chromosome number 11. And 2 genes are responsible for the 2 alpha genes are responsible for the formation of 2 alpha chains on chromosome 16. And a single gene on chromosome 11 is responsible for the formation of 2 beta chains. Okay, so look at this again here. Alpha, this is alpha gene family here. This is alpha gene family. Again, I repeat, there are 2 genes are there coding for alpha chain present on chromosome number 16. Along with these 2 genes, there is zeta gene is there, which is active during embryonic development. And the next one is, this is beta gene family located on chromosome number 11. Single gene is responsible for the synthesis of 2 beta chains, and it also contains one epsilon gene that is expressed in the early stages of embryonic development, and 2 gamma genes are there, G gamma and A gamma, which can synthesize gamma chains of fetal hemoglobin. And on beta family only, on chromosome number 11 only, there is a delta globin is there, which is found in adult hemoglobin HbA2. Okay, that is the basics of genes which are responsible for synthesis of globin chains. Now coming to hemoglobinopathies. Hemoglobinopathies already, as I mentioned, these are group of disorders which are caused by the synthesis of, which are due to the formation of abnormal hemoglobins or due to decreased production of normal hemoglobins, are rarely both. Okay, hemoglobinopathies are the group of disorders, maybe due to synthesis, okay, maybe due to formation or the synthesis of abnormal hemoglobins, formation of abnormal hemoglobins, maybe due to mutations, or else due to decreased production of normal hemoglobin, production of abnormal or due to decreased production of normal hemoglobins responsible for the diseases hemoglobinopathies. Now first let us discuss about hemoglobinopathies which are due to abnormal hemoglobin. Okay, one of the type of hemoglobinopathies is due to formation or synthesis of abnormal hemoglobin. Now here the first one is HbS hemoglobinopathies. First what we are discussing is it may be because of due to abnormal synthesis of hemoglobins. Okay, so abnormal hemoglobins, one is HbS. Okay, that is sickle cell hemoglobin. HbS is sickle cell hemoglobin or sickle cell anemia. Okay, now here this HbS, it is the most common form of abnormal hemoglobins. In the abnormal hemoglobins also most common one is HbS. HbS is nothing but sickle cell hemoglobin. Why it is called sickle cell hemoglobin? It is so named because the RBCs of these patients they will appear like sickle shape at low oxygen concentration. Okay, now here sickle cell here HbS is responsible for sickle cell anemia. HbS is an abnormal hemoglobin. Why it is called sickled hemoglobin? Because here in these patients RBCs they will appear like crescent like or okay, normal RBCs is like this due to the mutations. Okay, abnormal hemoglobin HbS has been synthesized. Here this one here the shape of this HbS is sickle shape, hence it is named as HbS. Okay, that is sickled hemoglobin. Now, the sickle cell anemia. Okay, so HbS is responsible for the sickle cell anemia. It is occurrence of this one is it is quite common. It is largely found in tropical areas of the world, and it is primarily occurs in black population. Now here, the sickle cell anemia is of two types, homozygous, heterogeneous. Okay, so SBS is responsible for sickle cell anemia. It could be seen in homozygous. It could be of two types, homozygous and heterogeneous. Homozygous means here it is caused by the inheritance of two mutant genes that code for beta genes. Okay, here mutation of the genes which are responsible for synthesis of beta chains. Two genes are mutated, one from each parent. So here homozygous is caused by inheritance of two mutant genes, one from each parent. Okay, so inheritance of two mutant genes that code for beta chains. And heterogeneous means here only one gene of beta chain is affected while the other is normal. Okay, here two genes are mutated in homozygous. In heterogeneous only one gene is mutated, the other one is normal. That's what here in heterogeneous 50% of the RBCs are normal hemoglobins, and 50% of the RBCs contain HbS. Because only one gene is affected in heterogeneous group of people, 50% of the hemoglobins are normal HbA, and 50% of the RBC contain HbS type of hemoglobins. So this is referred to as sickle cell trait. Sickle cell trait means 50% normal, 50% HbS. So these sickle cell trait people are asymptomatic. Okay, they can lead normal life. Okay, because all 50% RBC has normal hemoglobins, so they are asymptomatic. They can lead normal life. But in homozygous, both the genes are affected. Now here, molecular basis of hemoglobin S. So hemoglobin S, how it has been formed means normal hemoglobin we know that composed of 2 alpha, 2 beta. But hemoglobin S in sickle, okay, in this hemoglobin S, 2 alpha are normal, but there is mutation in the 2 beta genes. Okay, normal has 2 alpha, 2 beta in normal hemoglobin, adult hemoglobin. But in this abnormal hemoglobin S has 2 alpha, but mutation occur in 2 beta genes. Sorry, 2 beta chains. That mutation is due to, see here, this abnormal beta is due to single nucleotide. Okay, this mutation is due to single nucleotide. That means thymine has been replaced by adenine. Okay, So this abnormal beta change, the mutation which can replace a single nucleotide, that is thymine, has been replaced by adenine. Okay, so that means because of missense mutations, means if one single nucleotide has been replaced, so that is called missense mutation of beta globin gene. This causes the formation of altered codon, that is GUG, in place of GAG. So valine instead of glutamate at sixth position of beta chain. So that means here in the structure of so normal 2 alpha, but in hemoglobin A, if you look at the composition, the two beta globin chains are there. In each beta globin chain at sixth position,